Hi friends, today we are going to see the past year NEET questions for the chapter Respiration in Plants, and today in this video we will cover part 2. So let's begin with the first question here, which was asked in the year 2004. In glycolysis during oxidation, electrons are removed by dash. So which one will remove the electrons during oxidation? Now what do you mean by oxidation? How can you describe oxidation? See, oxidation can be described as a concept or a already we know we have reactions like reduction, oxidation. We even have a combination of redox reaction and all that. Now, oxidation can be described, or what happens in oxidation. If you see, oxidation occurs when an atom or a molecule or an ion will lose one or more electrons in a chemical reaction. Oxidation is a state of chemical species and it will increase. Oxidation doesn't necessarily involve oxygen. Originally, the term was used when oxygen caused electron loss in the reactions. Okay. So this is the meaning of oxidation. So you are given 4 options here. ATP. ATP cannot be the answer because that is the end product of a glycolysis. So that will not remove the electrons. Glyceraldehyde 3-phosphate, molecular oxygen. Once again I told you oxygen is not responsible for the removal of electrons. Even during oxidation, oxygen necessarily should not be there. It is not a necessity that oxygen should be present there. Okay. Therefore, molecular oxygen is also eliminated from the options. Now you have only 2 options left: glyceraldehyde 3-phosphate and NAD+. Now out of the 2 options, the correct answer is NAD+. Okay. Now, what does NAD stands for? Nicotinamide. Okay, nicotinamide adenine dinucleotide. So that is the full form of NAD. NAD+ is the answer. Okay. Now coming to the so ATP is the energy currency in all the cells. Glyceraldehyde 3-phosphate is reduced during. So what happens here, if you see, this is getting reduced during the process. So it is not involved in oxidation. Now NAD+, if you take this, molecular oxygen is a terminal electron acceptor. So molecular oxygen will help in electron accepting. Okay, electron accepting. And NAD+ is responsible for electron removal. Therefore, the correct answer to this question is option C, NAD+. And you have the process of glycolysis to your right side. So that is how the picture is. The cycle. First you have glucose 6-phosphate. It is converted into fructose 6-phosphate, then fructose 1,6-biphosphate. So the cycle goes on and the end product will be a pyruvate, and during during glycolysis, the net gain would be 36 molecules. Okay. So the correct answer is option C. Now let's move to the next question here. This question was asked in the year 2003. In alcoholic fermentation dash. So this is a very easy question for you. We already discussed in the earlier part, right? In alcoholic fermentation, the triose phosphate is the electron donor, with acetaldehyde is the electron acceptor. Apart from that, what happens in fermentation? Does it require an open system or a closed system? Did you ever think about that? See, fermentation is actually a chemical breakdown of substances by with the help of bacteria and yeast and other microorganisms, which typically involve effervescence, and that is produced by giving off heat. Okay. So fermentation, what is the process of fermentation? If you see, it's a metabolic process. Of course, the energy or the heat that is produced gives you some end product, right? It's a metabolic process in which the organism will convert the carbohydrates such as starch or sugar that we give in into alcohol or acid. For example, yeast performs fermentation to obtain energy by converting the sugar into alcohol. So this is the action that you see when you add in grapes and you add some yeast to it and you leave it undisturbed for many years, or example for like even 1 year. After that, when you take and see, it will be alcohol in it. You will find alcohol. The grape juice would have get converted into a alcoholic beverage called wine. So what happens here? If you see, the correct answer is option B. Triose phosphate is the electron donor while acetaldehyde is the electron acceptor. You need 2 categories there. Oxidation and reduction will take place, right? So you need an electron acceptor and an electron donor. Therefore, the correct answer to this question is option B. Very easy question for you to answer. Next question, which was asked in the year 2002. How many ATP molecules are produced by aerobic oxidation of one molecule of glucose? I think you know what the answer is. Okay, now this process, it is very easy for you to understand here. So what you have to know is, see, when they ask you the net gain, okay, net gain during this aerobic oxidation, it is 36. We all know that because we get 38 overall, but out of this 2 will be used for the link reaction. Okay, remember this here. And so the pending will be 36. So overall you have 36 in hand. This is what is the calculation. So here they are not asking you the net gain, but they are asking you how many ATP molecules are produced by aerobic oxidation of one molecule to glucose. So when the question is like this, they are not asking you what is the end product or how much we have in hand. They are asking you overall how much is produced. After that, how many are used or what is being used, they did not ask that question. They are asking us overall what is the production. Overall is 38. Now for example, we tell a class has 66 students. Okay, when someone asks you the strength of the student, you will only say 66. You will not tell who is absent, who is present, or on that day how many are present. All that information is not necessary. So here it is the same thing. They're not asking you how much you have in the last. They're not asking you the net gain. They're asking you the overall production. So the correct answer to this question is 38 ATP molecules. Okay, therefore the correct answer is option C. Next question here. Maximum usable energy per molecule of glucose metabolized will be generated during dash. So this was asked in the year 1999. So here they are asking us the maximum usable energy per mole of glucose. Metabolism, I told you, it is a very important characteristic of a living organism. Depending on the metabolism only, the energy of the animal lies. For example, we say that smaller animals have high metabolism, larger animals have low metabolism. What does a metabolism do? It actually releases energy. That energy is used by the organism to perform various actions. Even if you want to move an object, you need energy, right? Even if you skip one meal a day, you will understand how tired you are. Now, metabolism, all this resemble that. Now here they are asking the maximum usable energy per mole of glucose metabolized will be generated during which process? Okay, so last question only we saw about that. We spoke about aerobic respiration where 1 mole of glucose is used and it will yield about 38 molecules overall. 38 ATP molecules. So the hint is given here. They have mentioned it is per mole of glucose and it is metabolized will be generated during aerobic respiration by germinating seeds. Okay, so production of methanol, fermentation, all this will happen in the absence of oxygen, right? They are not aerobic respiration. So aerobic respiration is where glucose is involved and it will generate energy by metabolism. Okay, therefore the correct answer to this question is option A. The energy yield during aerobic respiration is 36, is 18 times of that during the anaerobic respiration, which is 2 ATP. Okay, next question here, which was asked in the year 1996. Okay, the enzymes hexokinase, which catalyzes glucose to glucose 6-phosphate in glycolysis, is inhibited by glucose 6-phosphate. This is an example of dash. See, you're given 4 options here, right? You're given 4 options here. Now, what do you mean by inhibition? How can you describe inhibition? Did you ever think about that? See, inhibition has a common meaning called feeling that makes oneself conscious and unable to act in a relaxed or natural way. So you're resisting something, right? Inhibition. Okay, what you meant, so you have different inhibition here: competitive, non-competitive, feedback, and positive inhibition. Positive feedback. Now what do you mean by competitive inhibition? How can you describe it? See, competitive inhibition can be described, or it's a process that resembles the normal substrate binds to the enzyme, usually at the active state, and prevents the substrate from binding. At the given moment, the enzyme may be bound to a inhibitor. So instead of binding, that binding process we are talking about here. Okay, so instead of getting binded to the usually at the active site, it what has to get binded. Instead of that, it will get bind to the inhibitor. Now, what do you mean by what do you mean by allosteric? That is very important, right? Allosteric inhibition. See, in biochemistry, allosteric regulation is regulation of an enzyme by binding an effector molecule at the site other than enzyme's active site. See, inhibition overall, if I have to tell you, I have to give you a clear idea of what is inhibition. You can describe it as a process which is not normal. Okay. Now, usually when an enzyme substrate complex occurs, the enzyme will go bind to the active site. But here it's just the opposite. Instead of getting binded to the the active site, it will get binded to the inhibitor site. So that is what is the overall thing that you need to know about inhibition. So here the enzymes hexokinase, which catalyzes glucose to glucose 6-phosphate in glycolysis, is inhibited by glucose 6-phosphate. This is an example of feedback allosteric inhibition. So you have allosteric inhibition and allosteric activation here to your right side. So in the allosteric inhibition, you can see the active site. So the active site gets altered here. So you can clearly see here, right? The active site is altered here. Can you see there is alteration? Now when it is activation, the substrate will correctly go bind to the, see, this is inhibiting the active substrate from binding to the active site. Therefore it will change the shape so that it cannot bind. Here if you see, it is correctly in the shape so that it can come and bind. Activation is the right process, that what happens in enzyme substrate complex. But here inhibition, it will avoid the substrate from getting binded to the active site. Therefore the correct answer is option C, feedback allosteric inhibition. Next question. This question was asked in the year 1996. Okay. Fermentation is anaerobic production of dash. I think this is a very easy question for you. I know by now you have all guessed the answer. Fermentation, as I already said, it is an anaerobic process which involves the yeast, microorganisms, bacteria which produce alcohol from carbohydrates or other substrates that you use in the process. Now fermentation is referred to as decomposition of organic compounds anaerobically with the help of the enzymes that the microorganisms will produce. So obviously it has alcohol, right? Apart from that, it will produce acid. So what is the right answer to this question? It is option B, alcohol, lactic acid or other similar compounds. Easy question, right? Next question. This question was asked in the year 1994 and again repeated in the year 1997. Fermentation products of yeast are dash. We so far saw about fermentation, right? What all it produces? It produces compulsory, it produces an acid. What all it produces? It produces an acid. Where am I writing? Okay. It is right. It produces an acid for sure, then alcohol. Apart from that, it also produces carbon dioxide because it is an anaerobic process. You don't have any whatever oxygen which is sustained in that container, that oxygen will be used, but there is no other presence of oxygen. Remember that the container will be closed, right? Whenever you do fermentation, the container is closed because there is no contact with the atmosphere. Therefore there is no production of oxygen. Therefore the correct answer to this question is option D, ethyl alcohol plus carbon dioxide. Okay, right? Predominantly it will be ethyl alcohol. Okay, ethyl alcohol can also be called as ethanol. Next question. This question was asked in the year 1994. In animal cells, the first stage of glucose breakdown is seen in. This is again an easy question for you. We all know when we talk about glucose breakdown, it always resembles glycolysis, right? So what is this glycolysis? I've given you the answer. The answer is glycolysis because we are talking about glucose. Now, how can you describe glycolysis? Glycolysis is a process of breaking down of glucose. We all know that, right? It's a breakdown of glucose. Glycolysis can take place with or without oxygen. Glycolysis will produce 2 molecules of pyruvate, 2 molecules of ATP, 2 molecules of NADH and 2 molecules of water. So where does this glycolysis take place? It takes place in the cytoplasm of the living cell. Okay. And how many enzymes are involved in glycolysis? Overall you have 10 enzymes involved in the process of glycolysis. Apart from that, what is the function of glycolysis? If you see, glycolysis is a cytoplasmic pathway which breaks down glucose into 3 carbon compounds and generate energy. Glucose will be trapped by phosphorylation, which will help the enzyme isokinase. And apart from that, you have ATP, which is used in the reaction, and the product glucose 6 phosphate will inhibit exokinase. Therefore, you will get the end product as pyruvate. And apart from that, you also have a net gain of ATP, which is in the form of energy where the body can use it. Therefore, the correct answer to this question is option B, glycolysis. It is the first step of respiration which occurs without the requirement of oxygen. So this is the only process which happens or which may not happen. It does not require oxygen. It can happen without oxygen, or if you have oxygen also, it's not a problem. The process can occur. Next question here. This question was asked in the year 1993. Okay. Complete degradation. So what is the question here? Out of 38 ATP molecules produced per glucose, 32 ATP molecules are formed from NADH and FADH2 in dash. Okay, this is a very easy question for you to answer. We know overall the net gain, when we talk about the strength, it is 38 ATP molecules produced per glucose. Now, 32 ATP molecules are formed from NADH or FADH. So what does this process denote now? It is obviously not Krebs cycle or EMP pathway. So it should be oxidative decarboxylation or respiratory chain. The correct answer is respiratory chain, because complete degradation of the molecule of the glucose will yield 38 molecules. So only in the respiratory chain, that is a glycolysis pathway, that is what is respiratory chain, okay, where you'll have 38 ATP molecules to be yielded. Okay, so when you have questions like this, they can have you asked in an indirect way. Now here they haven't mentioned it is glycolysis, but you have a hint which tells you it is glucose, right? It tells you, it has an hint that it is glucose. So it is very well understood that it belongs to respiratory chain pathway. Okay, next question here. So the correct answer is option A. Next question here. When one glucose molecule is completely oxidized, it changes to. Okay. Now look here. They're asking us a question which is very simple. They're asking us, one glucose molecule is completely oxidized. Okay, what does it give you? Now, complete oxidation of glucose will produce 38 molecules. We all know that, 38 ATP molecules. And that takes place in the presence of oxygen. So that you can call it as aerobic respiration. Now you're given 4 options here, right? 36 ADP into 36 ATP. No, that is not wrong, because we have only 38. So 38 will not be reduced into 36. Therefore, that is not the answer. So what is the answer here? The number of ATP molecules may, however, or may depend upon how NADH2 enters mitochondria. Okay, therefore the correct answer to this question is, say that 38 can, the variation can occur. Okay, so therefore it will be 30 ADP molecules into 30 ATP molecules. The correct answer is option C. Overall, we are not talking about the strength here. We are not talking about the net gain here. We are not talking about the part rest. What is after the link reaction? What is the procedure or what is the result? We are not talking about anything about that. Here we are just talking about one glucose molecule. When it is completely oxidized, it changes into 30 ADP molecules into 30 ATP molecules. Okay, next question here, which was asked in the year 1991. Out of 36 ATP molecules produced per glucose molecule during respiration. Again, this is an easy question for you. So this question here, if you see, out of the 36 ATP molecules produced per glucose molecule during respiration. Okay, so here you're given 4 options here. 2 are produced outside glycolysis and 34 during respiratory chain. 2 are produced outside is correct, but it is not 34 during respiratory chain. It is inside the mitochondria. Okay, 2 are produced outside mitochondria and 34 inside mitochondria. Okay, 2 ATP molecules which are produced during glycolysis in cytoplasm, while 34 ATP are produced in Krebs cycle. oxidative phosphorylation, and etc. Always remember that. Okay, therefore the correct answer is option B. Next question here, which was asked in the year 1919. End product of glycolysis is dash. This is a very easy question for you. So before we go into the answer directly, what is pyruvic acid? What is its use? Have you ever thought about it? See, pyruvic acid is a 2-oxo-monocarboxylic acid. What is it actually? It's a 2-oxo-monocarboxylic acid. Okay, carboxylic acid. Okay, that is a 2-keto derivative of propanoic acid. It is a metabolite obtained during glycolysis. It has a role as a fundamental metabolite and a cofactor. It is derived from which acid? Propanoic acid. Okay, always remember that it is propanoic acid. Okay, apart from that, what is the pyruvic acid in biology? If you see, it is a colorless, water-soluble organic liquid that is produced during the breakdown of carbohydrates and sugars during glycolysis. It has a chemical formula of CH3COCO2H. Okay, it is, if oxygen is available, pyruvic acid is converted into acetyl-CoA that enters the energy producing pathway, which is called as Krebs cycle. Therefore the correct answer to this question is option B, pyruvic acid. Next question. EMP can produce total. Now what is this EMP stand for? Do you have any idea? EMP pathway they will call it as. Now EMP is actually the other name for glycolysis only, the Emden-Meyerhof-Parnas pathway. So the name of the person is devised for this pathway. EMP pathway or glycolysis pathway, those mean the same. Okay, so don't get confused. EMP stands for, I'll write it down so that will be easy for you to understand. It is Emden, okay, Meyerhof, okay, Meyerhof-Parnas pathway. Okay, so the other name for glycolysis pathway. So I think by now you would know what the answer is. So here the net gain of the energy is equal to 8 ATP in the EMP pathway. Correct answer is 8. Okay. Next question here. Incomplete oxidation of glucose into pyruvic acid with several intermediate steps is known as dash. Again, this is an easy question for you. We all know incomplete oxidation of glucose into pyruvic acid. Where did we see all these components? Is it in TCA, HMP or Krebs cycle? It is during glycolysis. Glycolysis involves the process of partial oxidation. Where does incomplete or partial oxidation takes place? This is glycolysis. Okay, what happens here? A similar hexose sugar will break down into 2 molecules of pyruvic acid through a series of 10 enzyme mediated reactions. And we all know glycolysis occurs in cytoplasm of a living cell. What do you mean by incomplete oxidation? It is partial oxidation, right? The oxidation is not completed, right? So that is what is called as incomplete oxidation. Okay, so this incomplete oxidation of glucose, what happens here is not all hydrogen will be removed. What happens here? Not all hydrogen molecules will be removed. That is only the meaning of incomplete oxidation. Okay, the correct answer is glycolysis. Okay, next question here. NADP plus is reduced to NADPH is in which pathway? Again, this is a very easy question for you, which was asked in the year 1988. Complete degradation of the molecule of glucose produces 12 molecules of NADPH2. Okay, and this pathway or this belongs, or where it is reduced, if you see, it will happen in the HMP pathway. What do you mean? What is the full form of HMP? How can you call it as? You have other names for that. HMP shunt we will call it as. It's a pentose phosphate pathway. Okay, what is the pathway called as? Pentose phosphate pathway. It is a metabolic pathway that is parallel to glycolysis. What does it generate? If you see, it will generate NADPH. Okay, and pentoses as well as ribose 5-phosphate as a precursor for the synthesis of nucleotides. Okay, Apart from that, while the pentose phosphate pathway does not, it involves the oxidation of glucose, it is a primary role in the anabolic rather than catabolic. Therefore, the correct answer to this question is the HMP pathway, which is also called as HMP shunt. Option A is the answer. So with this, we complete part 2 of respiration in plants. So we will all meet in the next part. Okay. Thank you for watching my video.